Thank you so much, Dr. Gunn, for this fantastic talk. And next, please allow me to invite Dr. Samachari to lead the panel discussion. Perhaps to start from commenting on Roger's lecture.
Sure. Well, that of course was a beautiful exposition of all the quantitative data that you can extract for drug targets in the brain using kinetic PET analysis. So it's just amazing to see how much information you can pull. And I'm thinking about the fact a lot of that data was actually acquired on older scanners, and I think we're at an exciting point in the field right now where there's a lot of technology development happening that I think will further enable these kinds of studies. So I'm thinking about the fact there are new dedicated brain scanners being developed. We actually have 2 of the PIs of the 3 NIH-funded efforts on the webinar today. So we're going to see exciting new systems, and there are systems being developed in Europe and in Asia as well, as was reported at the IEEE meeting last week. So we're going to see a whole slew of new brain scanners with better spatial resolution, importantly, much better sensitivity, which I think will be greatly enabling for these kinds of studies. And then Roger also mentioned, of course, total body PET and long axial field of view scanners that open up the ability to look across the entire body kinetically simultaneously. And that's producing a lot of excitement as well. Improvements in time of flight on regular whole body clinical scanners is leading to great improvements in image SNR as well. So lots going on, and we can also perhaps debate the role of AI as well if we get a chance. So I think that's an interesting topic. So I'm really excited to see so many people joining these webinars. I think we need kinetic modeling in PET. It's really important that we don't lose this ability to quantify our signals and to further develop and apply these methods. I've been concerned that I thought it was a bit of a dying art, but I think there seems to be a renaissance right now, which is great, a lot of enthusiasm. And I'm noticing if I look across the people on this webinar, we've got some of the most senior people in the field that are on the call, I noticed, which is great, but we've also got a lot of really junior people here as well. And so that actually sort of leads me to the first point I wanted to bring up with Roger is, I think a lot of the people on this webinar come from academic backgrounds, and you've spent a lot of your career also in industry, Roger. And so I'm just curious if you can tell us a bit more about the role of kinetic modelers in an industry setting, both in a drug company such as GSK, and now perhaps tell us, I think a lot of people won't be familiar with who in vitro actually are and what they do and what their role is, because they're not a drug development company themselves. And so perhaps you can tell us a bit about that, because I think that'll be helpful from a career perspective for some of the junior people thinking where they could apply their skills.
Sure, I'm happy to mention a few things in that area. So I guess there's different places, as you indicated, academia, there's pharma companies, there's imaging CROs, so that's what in vitro does. The third one of those, we are an imaging CRO, so we provide imaging services for pharma companies, typically, in order to address some of the questions here. So we actually develop biomarkers, we deploy these imaging technologies, whether that be preclinically or in our imaging clinics in first time in human studies, single site studies, or run those multi-site studies that are being run out of 400 different imaging centers. So third one is imaging CRO. I guess you've also got to maybe not, it would be sort of imaging vendors themselves. So companies developing scanners and those types of things. So I'd say those were the sort of 4 areas where there are significant opportunities for kinetic modelers in terms of, to spend their days and do kinetic modeling, all slightly different. I think the thing to say about academia, you've obviously got the most freedom in terms of you can decide what you want to do, what you believe are the most important problems, if you can convince granting agencies to provide you the money to do it. In terms of pharma, you are very much aligned behind using imaging to advance the particular therapeutic programs that are being progressed by the companies. So depending on what company you work for, it may be that you need to show that imaging can impact on multiple sclerosis or something of that. So again, different people that will be directing where you go in that way. In terms of as in imaging CRO, we are aligned with developing imaging technologies and techniques that can impact on drug development as well, along with actually working with our sponsors to, jointly to have conversations and develop approaches to answer their questions, and really, can we develop things that allow them to develop go no go things. And then I guess the last one is the imaging sort of vendors, manufacturers themselves, and scanner manufacturers. Really, historically, they haven't really done anything significant in, I would say, in the kinetic analysis space. But with the advent of things like whole body kinetic imaging and things like that, I think we may even see them starting to change their minds and recognize. So I think there's opportunities in all four of those spaces for kinetic modelers, and there's pros and cons in each, and one needs to weigh those up to decide where you want to kineticize your life.
That's great. Thank you, Roger. So just for the audience, please enter your questions into the chat. I'm just a brief segue between Roger's talk and the questions, and I see we've already got a few, so I'll just ask Roger a couple more things, and then we'll switch to the audience questions. So an obvious thing from your talk, Roger, is that it's very brain focused, and it seems a pity to me that we haven't been using more of these approaches outside the brain. So I wonder if you can comment on that. Are the specific reasons it's much harder to look at protein targets outside of the brain, or is it just that there hasn't been that much interest outside the brain, and do you see that changing?
Really good question. I mean, I think in essence, there's no difference per se regarding protein targets. Everything just gets a little bit more difficult outside the brain. So you've got more challenges with motion, which becomes non-linear, as opposed to potentially a rigid body problem to solve. You've got metabolites as well. I would say those are the two big things. So often in terms of brain imaging, we design agents such that any metabolites are too polar to cross into the brain. So we're dealing with just modeling one species alone, if we get it right, for brain imaging. Whereas in the body itself, those metabolites, there's not those sort of membranes there that are going to keep metabolites out. So you're pretty much dealing with at least two species of different labeled molecule. And that just makes the modeling and the characterization that much harder, because you're typically only interested in one of those. So half of your effort is going to try and sort of remove, if you like, background or spurious signals before you can concentrate on what you're really interested in, and then try and estimate parameters and uptake from that key component. So I think those are the two biggest challenges in the body. But I think there's obviously a lot more information potentially coming from these whole body scanners, with the increase in sensitivity as well as the whole field of view itself. So that does provide increased opportunities to be able to do more sophisticated models that take advantage of multiple organs to try and to deliver on some of these difficult problems, particularly metabolite problems and things like that. So I think and the sensitivity shouldn't be underestimated as well. I think the dramatic increase in sensitivity really does offer many advantages. So let's see where people now take this new technology with these advantages.
So lots of opportunities outside the brain then. That's good to hear. And the final thing I just wanted to bring up was actually a couple of things that I didn't see much mention of that I wonder if there's a role. So obviously your talk was very PET focused, and we know the strength of PET and radio tracers for PET, but But what about SPECT? Is there much going on with kinetic modeling in the SPECT world? I saw you show one example of an intrathecal injection where there was a labeling with technetium-99m. So I was just curious, because I know there's a couple of SPECT people on the call at least.
I mean, it's not something that we're seeing a lot of activity in the in vitro space at the moment, other than with established technologies such as DAS imaging for Parkinson's, which is still the kind of go-to imaging readout for late phase trials. Although we are actually seeing AV133 probably, will start to displace that in coming years. So but there's still possibilities there. There's still, things that SPECT offers that PET doesn't. And I think it's all about, can you develop the right imaging agents? And that comes back to, probably touches on the previous question as well. This space is connected between great chemistry, great physics, great biology, and analysis sits on top of all of those things. So if we're labeling the wrong molecule in the first place, that makes the analysis really complicated. If we had bad scanners and actually the biological targets aren't relevant, then we're all going to, the analysis is very difficult and maybe not particularly of high value. So we need to get all of those things right and optimize them as well. To make kinetic modelers' life more straightforward, allow them time off now and again, sort of thing, but also to be pursuing these goals as a team. That's the important thing to recognize.
And the other emerging area I wanted to touch on is radionuclide therapy. So we're seeing obviously a huge growth in that area in oncology. And I wondered if there's any role for kinetic modeling there in the sense of, with the application of dosimetry in mind, and the fact that we're going to need to do better dosimetry in these patients, and there it's perhaps a case of having a model where we can extrapolate out beyond the imaging data to estimate the dosimetry accurately. So again, knowing the biology well, having a good model, having some imaging data to guide that. And any thoughts on whether that's a space that we could be working in?
So we're actually doing a large amount of work in there. There's a lot of companies that, I think we're going to see whether this is a bubble or not in the next couple of years. But at this particular point, there is a large amount of excitement and investment in this space. And I think modeling will be important to ultimately understand what is the right doses to be delivered to individual patients. So I think a real opportunity to, actually develop our models, make them more sophisticated, that they can potentially provide individualized plans for dosing for the subjects.
Great. All right, well, I think it's time to move on to the audience questions now. So I'll turn it back to Guobao so he can moderate the audience Q&A session. Thank you.
Thank you so much, Simon and Roger. So we got 4 questions from the audience, and we can answer it one by one. So the first question is from Yidan Wang. So Yidan, would you like to speak? If you like, you can also turn on your camera. You still have some time.
So this is a really great talk, Dr. Gunn. And my question is, do you think deep learning has the potential to accelerate drug development, and how would it combine with kinetic modeling? Thank you.
Great question. I mean, the first one's quite broad, and I will answer that to say to the point that the pharma industry is very excited about deep learning, whether that may need to be sort of moderated in certain cases. I think they believe it's going to solve most of their problems. I think deep learning is something that is going to be very good at solving certain problems, and it will solve them a lot better than we currently can. But it's not going to be the solution to everything. If we think about just ultimately what deep learning is, it's learning by examples. So actually, to do this really well, it needs lots of examples. So the problems where it's going to be incredibly effective is where there are lots of good, different, rich examples that enable it to really develop accurate models and include things that are unusual, because it needs to be robust enough to handle something that it wasn't expecting. You could have the best amyloid quantification algorithm and then somebody throws an FDG scan at it. So it's these things have got to be smart enough to determine those things. In terms of how does deep learning what does it offer in terms of kinetic modeling? So I think the jury's slightly out there in some ways. I think there's two ways to think about modeling. I think about what we call domain knowledge modeling and kind of machine learning as two different approaches. So you either build up models on the sort of machine learning side from examples, and on the domain knowledge side, you build models up from an understanding of the physics and the biology and the rest of it. And so we can think of today, what we would say is that we build up sort of compartmental models more from a sort of physics and biological understanding, and then we know we have characterization of things like ordinary differential equations and how that enables us to derive data from imaging. I think it's probably applying those domain knowledge models in certain frameworks that may be more robust to noise that may come from certain deep learning enhancements. But I think we may sort of see more in those settings, more hybrid type approaches rather than just blind deep learning approaches where we're just going to throw a load of examples at it and expect it to be able to work out what's going on. So I think if we, I guess to sort of distill that, if we have a lot of domain knowledge, we want to make sure that's encoded in our models whilst also taking advantage of some of these techniques.
Thank you, Dr. Gunn.
The next question is from Jose Anton. Dr. Anton, would you like to speak about your
Hi,
question?
can you hear me?
Yes.
Oh, thank you. Thank you for the talk and thank you for the opportunity. Yeah, my question is related to amyloid quantification. You've been showing a really exciting work around it, and it will be in terms of harmonization. When you talk about SUVR, do you use, for example, centiloid approaches, or do you use more specific target or and reference regions? I don't know if you could expand a little bit more on that quantification of amyloid using SUVRs.
So amyloid can be quantified, SUVR can be quantified in different ways in the sense that, but it comes down to choosing what is your target region and what is your reference region definition. In terms of looking at, I mean, and it's interesting in the sense that this is what the sort of spatial temporal modeling has kind of shown, that actually this stuff isn't changing particularly differently regionally, which is why many groups before we got involved in this work had determined that a big cortical VOI was the most sensitive target region for amyloid, and that the best reference region, if you were looking cross-sectionally, is gray matter cerebellum, and if you're looking longitudinally, is white matter cerebellum, which is interesting. So those things are sort of have been received and worked out sort of empirically from a lot of work. I think the interesting thing about the IQ stuff is it effectively produces a target VOI that is not dissimilar to a cortical one, but actually weights things appropriately across the whole image volume. And then the reference region that we obtain, if you think, which is really the non-specific canonical image, is actually something akin to white matter, which is not just restricted to cerebellum. And that's why we think we get additional signal to noise out of all of these data, is we're using all of the voxel data, we're taking advantage of that. And that's impacting on our increase in signal and power as compared to SUVR.
Thank you so much. Yeah, very interesting. Thank you.
Great. So the next question is from Dr. Gunn Wang. Dr. Wang, please.
Hi, Roger. Thank you very much for a wonderful talk. I have two questions. One is a practical one. Your equilibrium dialysis method is very innovative, but yeah, obviously it's quite time consuming for academic centers to do. So at least in the case of radio tracers that are already well behaved, if the plasma free fraction were obtained routinely in the presence and absence of an inhibiting drug, such as in target engagement, do you think it's necessary for us to have the free fraction in brain? And if it is, can you make public some of these equilibrium dialysis measured FNDs so that we can use them for current tracers? And my other question was direct versus the indirect estimation of multiple dosing from single dosing. I wasn't quite clear on the difference. So those are my two questions.
Nice to see you, Dean. Thanks for your questions. So the first question around FND, in terms of target engagement studies, FND doesn't change in terms of between baseline and post-dose studies. So it actually cancels out in terms of the calculation of occupancy as we can think about it. So we don't actually need a measure of FND for target engagement studies. The way we've used FND in terms of our work here is more in terms of looking at radio tracer development, in terms of predicting what the binding potential might be. So, I mean, the simple way for us to work in terms of identifying a new compound would be to say that we kind of know that a binding potential of about 3 or 4 is ideal. And we know that binding potential equals FND times Bmax over KD. So as a first port of call in terms of looking at new targets would be to understand what the FND of some molecules might be, what the Bmax is, and what type of KDs we have available to say, do we have something that's likely to provide a tractable signal? So that's one area where I think it is valuable to have FND type measurements. The other one is if you do want to get to a direct free concentration of your drug in tissue, then that can be valuable too. So I think, does that answer your first question?
So are there situations where you should measure the plasma free fraction before and after target engagement of a drug?
So in terms of if you're not able to get, if you don't have a reference region and you're looking at partition coefficients and total volumes of distribution, then it can be quite important to make sure you don't have significant changes in your plasma free fraction. So, I mean, we tend to always measure plasma free fraction anyway, baseline and post-dose, just to understand whether there is any significant change in that in the periphery.
So if you used an inhibition plot in that example without a reference region, and the free fraction changed, how do you address that? I'm talking about the free fraction in plasma.
Yeah, because I mean, if you're looking at inhibition plot, you're only looking at the changes in the total volume of distribution. Right. So you're not looking at the volume of distribution in relation to the free plasma concentration. So, you know, the ideal way to do it would be that you would have you would do your inhibition plots with VT over FP, and you would get the same thing if you just did them with VT, and you would go home and sleep at night.
Okay, thank you. And my second question, could you briefly clarify the difference between the direct and indirect method of predicting multiple dose from single dose?
So if you've got a direct relationship between which is typically the case for about 90, from our experience, it's about 95% plus of drugs have a direct relationship, which means that your target engagement can be described as, you know, an Emax relationship between target and the plasma concentration. In that case, you don't need to do a repeat dose occupancy study. You can calculate it just from your repeat dose PK concentration profile. You can just plug in the PK concentrations. It's only if you have this indirect relationship where you've got significantly increased target residence times and very slow kinetics then things get a little bit more complicated, and you need to sort of model the relationship with the more complex repeat dose PK profile.
Okay, and is the software for doing the indirect publicly available?
Not to my knowledge, but the differential equation is published, and I think you've got the skills to code it, Dean.
Thank you.
Great, thank you. We have the last question from Dr. Powell Markowicz.
Okay, hello, can you hear me?
Yes.
Hello, Roger. I just got a quick question, because you mentioned about this, particularly for the multi-center trials and on the dementia drugs and using the amyloid tracer, etc., and that you had this increased effect size. But because you use different scanners, like ADME, there's a huge variety in technology. And as Simon said before, we now have even better technology coming in. And obviously, the technology will not be replaced immediately everywhere. So we will have a mixture of different resolutions and different sensitivities. How do you think that is affecting the effect size? And would that improve the method, the amyloid IQ and tau IQ, if we had just, let's say, ideal one type of scanner and ideal imaging protocol, which would be identical everywhere?
So I think it would improve all methods, in the sense that, things that help things that should help any sensible method involve increased sensitivity, more counts, better statistics. If your method is not helped by that, it's probably not a good method. So I think, everything would improve in that line. Using just one single scanner that has the same geometry and blah, 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 should add to a little bit more consistency across subjects. But I think there's certain things that won't help. I think part of the strengths of amyloid IQ, in that sense, it comes down to the definition of both your target and your reference region in a smart way to maximize the information. So those are currently different in IQ versus sort of traditional SUVR methods. So I think whilst you would help both methods, I think you would see them both improve, and IQ still to outperform SUVR.
Thank you.
Great. I would like to invite Dr. Richard Castle to make some closing remarks. Rich?
Thanks, Guobao, and thanks, Raj, for a wonderful presentation, really demonstrating in interesting and very practical and important ways how modeling concepts applied in very different ways can really enhance what we can do and the impact of the data sets that we have. I think it's exciting, as Simon was alluding to, that perhaps, I don't know if it's quite a renaissance in modeling, but it's certainly something where the number of places where this can be applied continues to grow. And that's the fun part of this, that even using the same models with different tracers, different applications, different target organs, etc., what works and what doesn't work requires that careful evaluation. Sometimes we're lucky, everything works, we all agree, but more often than not, it takes a little bit of tweaking and understanding to make it work. I think with the advent of the new technologies, the high sensitivity systems, the total body systems, or the other dedicated brain systems that are coming, it's really going to open up that ability to take this to the next level. And there's a lot of different ways that could happen. Could be that we can extract better information, individual rate constants out of data. That might be one direction we can go. But I think something that will have more patient impact is just shortening the scan. Is that honestly, by shortening the scan, a lot of what we're doing in modeling is extrapolating to infinity. And if we can get that in a shorter period of time, we have better patient issues. We have less motion in those patients. And when we go outside the blood-brain barrier, we're going to have less chance for metabolites to get in and corrupt our signal. And so that big boost in sensitivity is something that's going to really let us jumpstart being able to apply these in more ways. And also, always the beginning of a scan gives us something about flow and function, and comparing that with our receptor targets or our protein targets, putting those two things together can only help us in terms of the kind of information that we can have. So very exciting stuff. Very cool to see the impact that in vitro others doing that work using the top of, state-of-the-art technology, what that's done for our field, and in terms of how it's being viewed in the pharmaceutical companies, et cetera. So thanks again to Roger for a wonderful, wonderful presentation over a lovely body of work over the years, which is very exciting to see that put together. Thanks so much.